So that's it. That's all persistent infections. That's the basic dynamic. So now let's talk about the strategies that different infectious agents use in order to ensure that they can persist. So one of the first strategies is that what the immune system is relying on to detect an infectious antigen are foreign proteins, that is the major antigens of the infectious agent. So what a lot of microorganisms will do, like the organism that causes malaria, is it will simply invade cells. So now it's inside the cells, that is, it's inside our own, in this case, red blood cells, and it's hard for the immune system to see it inside of those red blood cells. So this strategy of intracellular invasion is one of the probably most commonly used strategies that allows persistent infectious agents to actually set up these long-term infections. In contrast, most bacterial infections that our bodies clear, the microorganisms stay outside of our cells forever, and they're much easier for the immune system to find and destroy. But in this case, when the infectious agent's inside of a cell, it's a little bit more difficult for the immune system to recognize where exactly the microbe is. And this would be true for all viruses. All viruses, by definition, invade cells of our body. So not only is this true for the organism that causes malaria, it's also true for the organism that causes tuberculosis. It gets inside of macrophages and hides there. It's also true for Salmonella typhimurium, the thing that Typhoid Mary had. It invades cells of our intestine and sets up long-term infections there. All right, the second strategy, and basically both of these strategies are related. What's going on in intracellular invasion, the second thing, which I'm about to get to, is the organism that's hiding the things the immune system could see from the immune system so that the immune system can't recognize the infection. Another way to achieve the same end that's used by herpes viruses and Mycobacterium tuberculosis is they can actually turn off the synthesis of their foreign proteins. If they turn off the synthesis of their foreign proteins and the infection is quietly hanging out in one of these cells, the immune system also can't see it. So that's basically what periods of latency are all about, downregulating the thing that the immune system is looking for. It's effectively like the equivalent of a fighter jet flying so low that it's below the radar system and can't be seen. Same idea. So latency is an indefinite period of reduced or no protein synthesis that allows a microbe to become antigenically invisible to the immune system. No antigen means no detection by the immune system. Basically a game of cat and mouse. Third strategy, same basic idea, hiding antigens, but this one's used by bacteria like Klebsiella pneumoniae, Streptococcus pneumoniae. The protein portions of any microorganism are going to be the easiest thing for the immune system to go after. If you put a big, thick, sugary capsule around the outside of a bacteria, which is what a polysaccharide capsule is, it's much harder for the immune system to respond to that sugar coating. It's sort of like a cloaking device. Oops. All right, so basically first strategy, hide your antigens as well as you can from the perspective of the microbe. That's really going to mess up the immune system's ability to rapidly clear the infection. Second strategy is use DNA as your genetic material. That is, almost all the infectious agents that persist in our body use DNA as their genetic material for one very simple reason. DNA, unlike RNA, is incredibly stable. It lasts for years and years and years. Okay, so it may persist in the absence of active replication. The same thing is not true of RNA. So a lot of viruses are RNA-based viruses. Very few of them, there's only one I could think of, persist. All the ones that persist and cause persistent viral infections use DNA as a long-lived species to achieve that end. So, DNA viruses that persist, here's a few examples. Herpes viruses, all of them. All the retroviruses, hepatitis B, adenovirus, papillomavirus, molluscum contagiosum virus, which is related to the smallpox virus. And the only example of an RNA virus I could think of that persists is hepatitis C virus. But if you disrupt its replication cycle for a few months with, say, interferon alpha or antiviral drugs, that will actually get completely cleared from the body because the RNA of the virus is unstable. It can only persist if it's actively replicating. DNA viruses don't have that constraint. HIV is maybe one of the best examples of this. And HIV variant brings in RNA genetic material into the cytoplasm. It gets converted to DNA, and that piece of DNA gets integrated literally into your chromosomes. So you pick up an HIV infection, an extra gene, and this cannot replicate at all for 5, 10 years. As long as that cell stays alive, that latent HIV gene is sitting there. And at any time, it can reawaken and start making the products needed to make new infectious virions. In reality, people that have HIV, the cells that doctors are treating to bring down their HIV RNA load, probably about 1 out of every 400 HIV infected cells. The vast majority of cells are T cells that are latently infected with HIV. So even no matter how long someone's on anti-HIV drugs, you're never going to get rid of that huge reservoir of latent infection that can wake up at any time. Okay, back into the retrovirus life cycle. We don't have to talk about that. All right. So third strategy, changing your major antigens to avoid recognition by the immune system. And there's subtle versions of this, which is sort of the first one I'll mention. And the second one is sort of a very not subtle approach called antigenic phase variation. It's quite dramatic. So antigenic drift of HIV, what this basically means is that someone's infected with HIV, if they're not on antiviral drugs, the virus will continually mutate its proteins in a way that it avoids the current antibody response to the virus or T cell response to the virus. And it's basically slowly moving away from the immune response that's controlling it. And so those antigens change over time. In contrast, antigenic phase variation is an immediate and profound change in what an organism's antigens look like that give it a way to evade the immune response. So one example of antigenic phase variation would be in some bacteria they have one of the major things the immune response goes after, these long projections called flagella that allow the bacteria to move through the body. So if you make antibodies to these flagella, you can really slow down the spread of this bacterial infection. So in some species of Salmonella, basically they encode more than one flagella. And that's when they switch from one flagella to another flagella that's antigenically distinct. That's called antigenic phase variation. So the way that works, to give you an idea, is you'll have a promoter region that drives expression of two genes called FLAG-B and FLAG-A. FLAG-A is a negative regulator of another flagellar gene. It keeps it off. And FLAG-B is the flagellar protein or flagellar protein that the bacteria uses to swim around. This promoter region driving the expression of these genes can actually invert. It's got two sequences that allow it to be transposed or inverted. And when that happens, the promoter becomes inactivated, FLAG-B is no longer made, the repressor of Fly-C is no longer made, and Fly-C makes a second flagella. So basically, in a population of bacteria where antibodies are really going after all the organisms, they have FLAG-B. If this thing switches to making Fly-C, it's a rarity, it might only happen in 1 out of every 10,000 bacteria, but the antibody response totally misses this second related organism and it takes off again. The place where this idea of antigenic phase variation is more extreme is there's some species of Borrelia bacteria related to Lyme disease that can undergo many antigenic phase variations. And probably the most extreme is this trypanosome that causes African sleeping sickness. The organism looks like this. The main thing to appreciate is that on the outside of the organism, there's something called the variable surface glycoprotein. And this organism basically has a suite or a bunch of cassettes of different variable surface glycoproteins on the order of hundreds to thousands of different variable surface glycoproteins. And so what happens in African sleeping sickness is somebody gets bitten by the microbe or by the vector, that is the insect that carries this microorganism. It gets in your blood and it causes basically a relapsing fever. That is, if this is the microbial burden shown here as a blue line, one particular variant expressing this variable surface glycoprotein starts to increase in number, and it decreases when your immune system starts to control it. As the antibodies come up, the load goes down. But then it swaps out this gene for a new gene. And the organisms that display this new gene completely get around the antibody response you just made to the old organism, and it starts to go up. And it starts to come down when antibodies to that variable surface glycoprotein come up, and that happens over and over again. So if somebody has this sickness and is untreated, they will eventually die, because the organism just has that many different antigens where it can basically every 2 weeks cause a new recurrent fever. So basically, antigenic phase variation is the equivalent of antigenic shift that happens in influenza, but happening from one organism within a single host. All right, so we've talked about 3 strategies to get around the immune system. A 4th strategy is to colonize sites in the body that are very difficult to attack. Maybe the most common example of this is tuberculosis. The microorganism is inside these things in the lungs called granulomas. It gets walled off by the immune system in the form of granuloma. And it's a compromise. The immune system has walled this off and contained it, but the microorganism lives in here happily for years, decades. And at a later point in time, particularly if there's injury to the lung, it can come back out and cause tuberculosis again. Likewise, there are these tapeworms that the species is called Echinococcus, or the genus is called Echinococcus, and they form basically little cysts inside of the tissue that are impossible for the immune system to go after. And so it just lives in there happily. And when they're removed from the body, here's what they look like. So 4 strategies colonize sites the immune system can't reach. The 5th strategy, and we'll be hitting upon this more later, is active obstruction of the immune effector mechanisms. That is, there are a set series of antibodies, T cells, that is a set number of attacks that we use to control microorganisms. And some of the microorganisms, like Staphylococcus aureus, have learned this. And so they make proteins, such as protein A, that basically completely derails what your antibody response is trying to do. This is called immune evasion, the active interference with the way the host immune system normally works. And what it does, it renders Staphylococcus aureus very difficult for the immune system to prepare for food that is opsonized, so it can be eaten by neutrophils. As a result, Staph aureus tends to not only form, pus-forming boils under the skin, but once they're there, they can keep coming back, every 2 or 3 months, because it's just that hard to clear the infection completely. All right, so those are the basic strategies. We just talked about them. Like I said, I'm going to go back, we're going to be revisiting the idea of immune evasion as we move forward in the lecture. But those are 5 strategies that you could use to think about how any infectious agent persists in the body. All right, so now let's focus in on the alpha herpes virus as our model organism to think about persistence. And we're going to talk basically for the remainder of the lecture about just this one group of viruses. First of all, I want to introduce the alpha herpes viruses to you and explain what they are. I then want to talk about how the immune system controls viral infections. You have to understand that first before you can understand what the virus is doing to derail that process.